Welcome to Food Science Peeps. In the last video, I've explained 5 water-soluble vitamins, including their names, structures, general properties, biological functions, excess and deficiency, food sources as well as stability. In this video, I'll continue with another 4 water-soluble vitamins. Now, let's start with vitamin B7, which is also called biotin. Biotin consists of an uriato ring fused with a tetrahydrothiophene ring. The tetrahydrothiophene ring has a valeric acid attached to one of its carbon atoms. Biotin obtained from dietary sources exists in both free and protein-bound forms. Biotin has 3 asymmetric carbon atoms, thus has 8 different stereoisomers. However, only D-biotin has biological activity. Now, I'll talk about the biological functions of biotin. Biotin is important in energy production by supporting enzymes involved in metabolism of fat, carbohydrate and protein. Biotin plays a role in many metabolic pathways such as gluconeogenesis, fatty acid synthesis and breakdown of amino acids. Besides, biotin is important in maintaining hair health. Although there is no strong evidence to prove that biotin increases hair growth and strengthen the hair, but deficiency in biotin results in hair loss. Apart from that, some studies show that taking biotin supplement can lower blood sugar levels in some individuals with type 2 diabetes, especially when taken together with chromium. Next, excess and deficiency. Toxicity caused by biotin is rare because human bodies can tolerate high doses of biotin. However, excess biotin accumulation can inhibit endogenous sirtuin activity. Sirtuin is a class of proteins that regulate cellular homeostasis. Thus, excess biotin accumulation may affect cellular homeostasis and cause negative effects such as increased inflammation, cellularity, and collagen deposition. On the other hand, not enough of biotin can cause neurological symptoms such as depression, lethargy, hallucination, and numbness of hands and feet. In severe cases, lack of biotin can lead to alopecia, which refers to hair loss from part of head or body. The severity of hair loss can vary from a small area to the entire body. Serious deficiency in biotin also causes dermatitis. The symptoms include scaly, red rash around the eyes, nose, mouth, and genital area. Next, food sources of biotin. Biotin can be found in many foods. Foods contain the most biotin are organ meats, seeds, cheese, eggs and vegetables such as spinach and sweet potato. Next, stability. Biotin is stable to heat. Heat treatment at 100 degrees Celsius in moderately acidic and neutral condition doesn't cause significant loss of biotin. However, its stability decreases above pH value of 9. Vigorous treatment with either acid or alkali can destroy biotin, but biotin is more unstable in alkaline condition compared to acidic condition. On the other hand, biotin is not sensitive to oxygen even in acidic and alkali condition, but it is destroyed by strong oxidizing agents such as hydrogen peroxide and bromine water. Moreover, biotin is sensitive to light, especially to ultraviolet light, which can cause degradation of biotin. Next, let's learn about vitamin B9, which is also called folate. Folate consists of 3 subcomponents, which are pyridine ring, para-aminobenzoic acid, and glutamic acid. Folate has another form, which is called folic acid. Folate is the natural form found in plant-based foods such as asparagus, lentils, and peas, whereas folic acid refers the synthetic compound normally added to fortified foods. The difference is folate is in reduced form while folic acid is in oxidized form of vitamin B9. Although there is a little bit difference, but they have same biological effects. Now, let's look at biological functions of folate. Folate is needed in synthesis of genetic materials. 5,10-methylene tetrahydrofolate is a compound formed from folate derivatives. It serves a cofactor in many biochemical reactions. One of its important activities is, it donates a methyl group to uracil, converting it to thymine, which is used in synthesis and repair of DNA and RNA. This is crucial in cell and tissues growth, especially during infancy, adolescence, and pregnancy. Besides, folate is important in erythropoiesis, which is the production of red blood cells. Folate works together with cobalamin in producing normal red blood cells and helps iron function properly in the body, thus preventing megaloblastic anemia. Apart from that, folate is important in preventing age-related health problems. For example, methylfolate, a derivative of folate, is essential in the brain for making neurotransmitters that are important for maintaining cognitive function, preventing dementia. Next, excess and deficiency. Studies suggest that adequate folate intake can reduce the risk of getting cancer. However, too much folate can also stimulate growth of cancer cell due to DNA hypermethylation. Folate blood level is associated with methylation of tumor-suppressing gene and development of cancer cells. Thus, excess of folate can lead to the increase in cancer risk. Besides, supplementing with too much folic acid may increase insulin resistance. This can result in disruption of glucose metabolism, which is the driving factor of type 2 diabetes, gestational diabetes, and also prediabetes. On the other hand, deficiency in folate may cause neural tube defect, which affects spinal cord, brain, and skull of newborn baby. During pregnancy, folate is important to produce blood cells, DNA, and help the growth of baby. Neural tube defect happens due to malformation of tissues and bone around the brain and spine, which is associated with inadequate folate intake of pregnant women. In severe case, this may cause stillborn or neonatal death. Now, let's look at foods rich in folate. Folate can be found in vegetables, especially dark green leafy vegetables such as spinach, lentils, asparagus, and broccoli. Other food sources include meats, poultry, grains, and eggs. Next, stability of folate. Folate is one of the most unstable vitamins. Folate is relatively stable at temperatures lower than 180 degrees Celsius. Upon 180 degrees Celsius, folate starts to degrade. Research shows that folate becomes more stable to heat in the presence of ascorbic acid. Besides, folate is susceptible to oxidative degradation in the presence of high oxygen levels. Ascorbic acid also helps to prevent oxidative degradation of folate due to its antioxidant properties. Moreover, folate is stable in acidic condition but becomes easier to degrade as pH increases. Rapid destruction of folate occurs when heated in neutral or alkaline solution. Folate is sensitive to light, especially the ultraviolet light. UV light will degrade folate by cleaving the methylene bridge. Next, let's look at vitamin B12, which is also called cobalamin. It is the largest and most structurally complex vitamin. It consists of a corrin ring with cobalt at its center, which is linked to the nitrogen from 4 pyrrole groups. Besides, there is a ligand linked to the cobalt called 5,6-dimethylbenzimidazole. This benzimidazole is linked to a 5-carbon sugar, which is attached to a phosphate group. The cobalt has already formed 5 bonds with other atoms. The remaining 6th position is the active site, which allows the cobalt to link other ligands, forming different chemical form of cobalamin. For example, it is linked to a methyl group to form methylcobalamin, it can form cyanocobalamin when it is linked to a cyanide group, and so on. Now, let's look at the biological function of cobalamin. Cobalamin is essential for the production of healthy red blood cell. During erythropoiesis, which is the process of producing new erythrocyte, erythroblasts require vitamin B12 for proliferation during their differentiation. Besides, cobalamin also plays an important role in maintaining nerve cell functioning. Studies show that vitamin B12 promotes nerve repair by speeding up nerve tissue regeneration. It also reduces inflammation, thus contributing to the health of nervous system. Another biological function of cobalamin is metabolism of fatty acid and amino acid. Research shows that one of the chemical form of cobalamin, called adenosylcobalamin, acts as the coenzyme for enzymes involved in catabolism of fatty acid and amino acids. Next, excess and deficiency. Cobalamin is a water-soluble vitamin. It can be excreted through urine. Thus, it is considered safe and has low toxicity even at high doses. However, high-dose injections of cobalamin can result in some negative effects. For example, outbreaks of acne and rosacea, greater risk of heart attack, stroke, and death. Deficient in vitamin B12 can lead to pernicious anemia, which is a chronic disease due to abnormally large red blood cells. Lack of vitamin B12 will inhibit purine and thymidylate syntheses, impair the DNA synthesis, and cause erythroblast apoptosis. This eventually results in anemia due to ineffective erythropoiesis. Inadequate intake of vitamin B12 may disturb the nervous system and cause neurologic symptoms such as paresthesia in hands and feet, muscle cramps, dizziness, cognitive disturbances, ataxia, and erectile dysfunction. It also leads to psychiatric symptoms such as depression and drowsiness. Now, let's see what foods are rich in cobalamin. We can get cobalamin from liver, lean meat, dairy products, fish, and shellfish such as salmon, clams and crabs. Next, stability. Cobalamin is sensitive to light as it degrades in the presence of light. Research shows that degradation of cobalamin is higher when exposed to sunlight compared to artificial light. Cobalamin is stable at the pH ranges from 4 to 6.5. It is very sensitive to strong acids and alkalis. Cobalamin is stable to heat as no significant loss after heat treatment such as pasteurization and cooking. Some studies show that ascorbic acid can decompose cobalamin in laboratory conditions. However, there is no sufficient evidence showing significant destructive effect of ascorbic acid on cobalamin in human bodies. Moreover, cobalamin is bound to various proteins in our bodies such as transcobalamin, thus preventing itself from destruction by ascorbic acid. Cobalamin in food, on the other hand, is bound to proteins that protect it from decompose. Next, let's look at the last one, vitamin C, which is also known as ascorbic acid. Ascorbic acid has an enediol structure conjugated with carbonyl group in the lactone ring. The two enolic hydrogen atoms are responsible for its acidic properties. Ascorbic acid is readily reversibly oxidized into dehydroascorbic acid. The dehydroascorbic acid has almost the same activity as ascorbic acid. Now, let's look at the biological functions of ascorbic acid. Ascorbic acid acts as an antioxidant. It protects carbohydrates, proteins, lipids, and nucleic acids against the free radicals damage. This is because it can donate electrons from the second and third carbon to neutralize the free radicals and terminate oxidation chain reactions. Ascorbic acid is also needed to enhance the absorption of iron. Ascorbic acid can reduce the inorganic ferric into ferrous form, which is easier to be absorbed by our bodies. Heme iron is absorbed more easily by our bodies compared to non-heme iron. Hence, adequate vitamin C is especially important for the absorption of non-heme iron, which is normally found in plant-based foods. Apart from that, ascorbic acid plays a significant role in the formation of collagen. Collagen is a protein that acts as primary structural component of connective tissues, such as skin and cartilage. Ascorbic acid acts as a cofactor for the hydroxylation of proline and tyrosine, which are the amino acids needed in collagen synthesis. Next, excess and deficiency. Toxicity of vitamin C is very rare because our bodies cannot store it. However, vitamin C intake more than 2000 mg per day can lead to nausea and diarrhea. On the other hand, deficient in vitamin C can cause a disease called scurvy, which is uncommon but still occurs in developed countries. The symptoms include weakness, fatigue, sore arms and legs. If left untreated, more severe symptoms like decreased red blood cells, gum disease, hair loss, and bleeding from the skin may occur. Next, food sources of ascorbic acid. Citrus fruits are the most famous food sources for ascorbic acid. For example, lemon, grapefruit, and orange are all rich in ascorbic acid. Besides, we can also obtain ascorbic acid from vegetables such as broccoli, bell pepper, tomato, and many more. Next, stability. Ascorbic acid is readily and reversibly oxidized to dehydroascorbic in the presence of oxygen. This can be affected by many factors such as temperature, pH, concentration of oxygen, and presence of heavy metal. Besides, ascorbic acid is also sensitive to light. Exposure of foods rich in vitamin C to UV light, orange juice for example, causes more than 15% loss of ascorbic acid in the juice. Ascorbic acid is chemically stable in acidic condition but unstable in neutral and alkaline conditions. Apart from that, ascorbic acid is sensitive to heat as it starts to decompose at temperature as low as 30 degrees Celsius. Heat treatment of foods rich in ascorbic acid at 30 degrees Celsius to 60 degrees Celsius results in the conversion of ascorbic acid into dehydroascorbic acid. However, same treatment at 70 degrees Celsius to 90 degrees Celsius retained vitamin C as ascorbic acid due to the inactivation of the enzyme ascorbic acid oxidase by heat. Conversion of dehydroascorbic acid into dictogulonic acid causes it to lose biological activity. Hence, it is suggested to use heat treatment above 70 degrees Celsius to prevent conversion of ascorbic acid into dehydroascorbic acid. That's all for today's lesson. Thanks for watching, bye.